the three speakers are here, and we're happy to take any questions if you have any. I would just, if there's one takeaway that I would ask for each of you, is not that PKPD is super important or anything like that, because I understand that a lot of you work in completely different areas, but it's more so about trying to explore every element of a research study that's possible. And so performing all of these smaller sub-studies to try to understand why you might see the effects that you do in, it could be a clinical trial, it could be, some other study that you're doing as well. Things such as implementation science, qualitative evaluations of consumer or patient experiences with an intervention, which could be a treatment, could be a diagnostic. These are all really important aspects to consider in project design, which can really facilitate the next phase of research for your program. And so I just ask you all to consider very carefully about how you can be designing your own projects such that you can continue to optimize whatever that treatment or diagnostic or whatever it may be that you're developing, such that that program can be sustained and be maximally valuable. Maybe is there a way that we ask if there's questions somewhere else?
Do you have any question in Dotton Park or Helston? Or even if you want to talk in Dotton Park, you can turn on your microphone. So we have a question here.
Hi, I just had a question for you about the antifungals. So you were looking at critically ill patients, but what's the standard therapeutic dosing for a normal patient, like compared to what you saw in those different sites and, that final data? So what's, the disparity between those two?
Between critically ill and standard?
Yeah, so what would normally be administered for a standard patient? So you're looking at that critically ill cohort. I just wondered, because there was a huge diversity I saw in the dosing that was being administered. So I wondered, what's the normal? So what would be, or there isn't such a thing?
I think that's a hard question, because the clinical guidelines the different sites stick to are different because of so many different countries. So what France might consider standard might be not standard in Australia. Although they do have, I don't know how to explain this.
Like the pharmaceutical company, I assume, is telling them to go with a certain dose, or that's, I suppose, what I'm trying to
I understand.
Sorry.
The question. No, the dose are a standard dose that is given to a patient usually comes from phase 3 trials where they have identified that, okay, this dose is going to cause the best effect. And that's how a dose is established. Yeah. However, they usually come from a more general, not healthy, a more general population. And that's just what's given to everyone. But what people had noticed is that this might not be working critically ill patients. So that's what we were trying to address. And we did find that, nope, it's not working for some of them. Some of them are performing well, but there's areas where another antifungal should be looked at in this population.
Jason, can I make a comment? Can you hear me?
Oh, fantastic. Emeritus Professor Jeffrey Lippman, the Godfather. We would love to hear your opinion.
So we describe, to answer
Amanda's question.
Yeah, to answer that question, I just go back to many years ago, we described in ICU a phenomenon called augmented renal clearance. Now, what that came from is the result of the cause of the augmented renal clearance is you have an inflammatory response. Your body has a blunderbuss effect of saying there's a problem, I'm going to give you more substance, which is the only thing the body can give you is more cardiac output, higher blood flow. So you have an increased blood flow throughout the body, increased renal blood flow, increased glomerular filtration rate, and higher excretion of all renally excreted drugs. So that's been well documented. And that occurs with the beta-lactams that Hafiz mentioned and most of the antibiotics. The antifungals are more difficult to work out, partly because a lot of them are hepatically metabolized. And when you have an increased blood flow to the kidneys, you'll have an increased blood flow to the liver as well. Now, you can't measure that as easily as you can renal function. So what I think is happening, and I was going to bring it up with Paddy's talk, I think what's happening with the antifungals is there's a higher cardiac output, higher hepatic blood flow, and therefore increased clearances from ICU patients for the antifungals. It just hit me when Patty was talking that we haven't been able to measure hepatic blood flow, but I think that answers the question why the ICU dosing is not the same as a package insert, because the drug companies aren't going to do the pharmacokinetic studies in ICU. It's too dangerous, too many people die, and the drug will fall over. So I think that answers some of the question, and it just hit me when Patty was talking. Thank you.
So, Jeff, can I just add to that? This is Mike. What about ICG and just using that little tiny finger monitor to know what's going on with liver blood flow?
Well, we haven't done it. You can do it, but it's, you need a formal study for that, but yes.
Because Peter Kruger's got some stuff on that. We were doing that at PA. You may want to, I think we have the gear for it too, if you need it.
Yeah.
I hate to burst your bubble, guys, but there's a high clearance and low clearance drugs through the liver. And if it's a high clearance drug, then certainly you're right.
Yeah.
But a lot of these are going to be dependent on metabolic capacity of the enzymes, and drug interactions is probably something which is a bigger issue that's occurring here. And I think organ dysfunction more than enhanced organ function, just with the nature of fungal diseases. That's why fluconazole, I think, does relatively well in this cohort, because we actually do have ICU specific dosing. But we've done studies with ICG before, as directed by you, Emeritus Professor Michael Roberts, with Emeritus Professor Jeffrey Lippman. And it certainly is something which is very feasible to do in pharmacokinetic studies. And the only way you're going to test these things is if you actually do the study. So that's great advice. Thank you.
Just before you leave that, though, you can also measure that metabolic function. Presumably you're doing that, are you?
There's only so much we can do. We're measuring the drug exposure and whatever else we can. As you know, we have tried to include some marker compounds to describe some physiological functions which are likely to affect pharmacokinetics, but sometimes you don't know until you do the first evaluation, which is where we're currently at with the antifungals. Ninka.
Very impressive collaboration across the world. Just curious, with the Bling study, I noted there was quite a large group of people who didn't consent to their data being shared. Do you know what the reasons are for that? Or
so there is in the consent form there are two elements: the Bling study and also there are two elements for the Bling study and Bling PKPD. So for Bling PKPD, there were several additional processes like blood collection, isolates collection that would be performed. So if they don't agree to that, they don't agree with the additional processes like blood samples, isolates, so they can in the consent form tick they don't agree. So maybe I think you're referring to that for the PKPD sub-study. Yep.
So thank you, everyone. I think we are running out of the time. That was a very amazing presentation from all the speakers. Thanks a lot again. And yeah, so next week we're going to have another seminar series, and hope everyone enjoyed that. I'm sure everyone enjoyed. Bye for now.